You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.
Hello, this is Richard Jacobs with the Future Tech and Future Tech Health Podcast. I have Semir Beyaz. He holds a PhD from Harvard. He has a lab at Cold Spring Harbor Laboratory, very famous lab for many years. He's principal investigator there, and we're going to be talking about cellular signaling and cellular states. So I'm looking forward to this. So Semir, thank you for being here.
Thank you.
Yeah, so tell me, how would you describe your work? What would you call it? I just gave a very brief description, but what are you working on?
Yeah, so the simplest question we are trying to address, which is actually very complicated, is to figure out if we are really what we eat. So diets can play a huge role, as you all know, in our lives, including contributing to disease states such as obesity and cancer. And there's a lot of ambiguity in terms of the signaling mechanisms or the cellular mechanisms that can influence how nutrients are regulating our physiology at like organ level, molecular level, and cellular level.
And so, traditional medicine sadly seems to say that nutrition really doesn't affect biology very much. But when you look at it, let's say we're going to live 80 years. The only inputs to our cells, the primary inputs, are food, water, air, then maybe chemical exposure, etc. Just to me, it's like, how could those not be the major defining inputs of what we are? We're made up of what we consume.
Yeah, you're absolutely right. I mean, even if you look at the most simple organisms, what they eat define their status, and their health is dependent on that. So we are not different than this. We are significantly shaped by what we eat. So diet and metabolic alterations due to diet can have a significant effect on physiology, on the way that organs function. The cells within that organs are influenced by that, and microbes in our guts that can influence many physiological processes are significantly dependent on what type of nutrients we are consuming. So you're right, actually, the nutrients are significantly impacting our lives, but we know very little about how they are doing it.
Even, I was thinking, even the act of eating, it's weird if you think about it, like literally what's happening. In one way, I don't know, maybe I'm wrong about this, like our physiology is using the bond energy of the electrons and the molecules of the food we're eating to power ourselves. In another way, I'm sure we're taking in the constituent atoms and molecules that constitute the food and literally integrating it into our structure ourselves. But when I thought about eating, I mean, is that what's going on? Or am I crazy? And if so, it's weird to think about it that way.
Yeah, I mean, the main reason that we eat is we need energy. So it's understood in that context, like how you digest nutrients when you eat, micromolecule digestion is, we understand how our digestive tract is handling these nutrients. So let's say if you eat a piece of cake, we know what it is going to be converted at the micronutrient level, such as sugar, amino acids, or fatty acids. We know how these are processed in the body, and that was understood the past century. However, we have very limited understanding what happens next. So let's say you consumed a burger, or you ate bacon, and then that had lots of fat in it, and that fat get digested into fatty acids, and then that fatty acid now is circulating in your body or being stored in some cells. How this fatty acid now is influencing those cells that it is exposed to. And so this is one research question that we have in our lab. We try to understand how dietary fat and obesity, which can have dysregulated fat biology in the body, can influence diseases such as cancer. And epidemiological data suggesting that obesity is strongly associated with diseases like cancer and type 2 diabetes, but the causal mechanisms are still not very clear. So in the lab, that's the link that we are trying to make. From the moment you eat something, how does it transform your body at the physiological level, cellular level, and molecular level? And I can give you more information if you are curious as to specific examples.
Yeah, right, exactly. So you're in one way you're studying eating, like literally what is happening when you're eating. Like I said, we have something called the electron transport chain. It's part of sugar metabolism. So I mean, in one way, literally, it's like it's kind of like the Panama Canal. We're using literally the bond energy of various molecules and moving this one here and moving that one there, and are we actually literally using that energy of those bonds to power us, or we're using that energy of those bonds to assemble molecules that are beneficial to us, to break down other ones. I mean, it's just a strange thing when you think about it.
Yeah, so both is true, what you're suggesting. So we use and what we eat influences how this energy is being utilized in each cell in our body. But in addition to that, what you eat can also provide some input to each cell to instruct that cell what to do. So those are like the environmental signals that regulate or that help a cell to function or to acquire a phenotypic state. So I'll give you an example. For example, if you eat a nutrient that has a lot of vitamin A, that vitamin A can carry out some vitamin A related metabolic functions, and also can be a ligand, can be a signal for a specific receptor in the body that can activate and turn on certain genes in response to vitamin A. So you both use nutrient as a source of energy where you incorporate this into your body, and it can be part of your body mass, or it can be used as an energy source to carry out certain reactions, but it can also be used as a signal, as an environmental signal in your body.
So, what I don't know, what amazes you about what you're studying? What surprises you? What new things have you figured out that you can't believe it?
Yeah, so we initially focused on a model where we try to understand how diet can influence the risk of cancer by feeding certain mouse models with high-fat diet and try to assess cellular and molecular mechanisms that can contribute to cancer risk. And we found, very surprisingly, that the fatty acids that are in the diet can activate stem cells in an abnormal way. And those stem cells, when they are abnormally activated, they can acquire some oncogenic mutations and contribute to cancer. So now we are expanding on this observation and trying to figure out how these individual fatty acids can affect immune system, can affect microbiota, can affect the way that immune system can survey cancer cells. And we are finding lots of interesting mechanisms that can help us to treat cancer either in the context of obesity or, just in general, by utilizing immune system against cancer, which is now very common with the discovery of, immunotherapy as a potent therapy for cancer.
Well, first question. So you said you fed rats a high-fat diet, but high-fat and low-carb, or just high-fat period, high-fat and low sugar? I mean, what were the other? What was the other constituency of the macronutrients?
Yeah, so this is a very commonly used, the initial study that we used was a very commonly used diet-induced obesity model, which is high in fat and also not low in carbs. So it was, I would say, like 60% of the calories were coming from fat, but it had also enough macronutrients in the context of like carbohydrates. However, you are touching on a very important question. How does diet pattern, like when you change the fraction of other nutrients, how does this influence cancer incidence or biology, is an active area of investigation. So if you have very low carb with very high fat, that's called ketogenic diet, because you force your body to produce molecules called ketone bodies when you don't have enough sugar to use as a primary energy source. So a high fat, low carb diet does not always have the same effect as a high fat, high carb diet. So that's the complication. We, by changing the ratio of two macronutrients, you can change drastically how these cellular pathways are changing that can influence the risk of cancer.
Right. Well, so I think it's important to point that out, because then you'll say, oh, a high-fat diet leads to cancer. Well, not necessarily. It could be high-fat in the presence of high sugar, the presence of carbs, not in the presence. So why is that so common in scientific studies where that's not elucidated? Because it's very misleading, in my opinion.
Yeah, I mean, so that's basically the rationale for us to study that, because, there is not much being done to assess the causality of these associations. And so, we published some papers showing some pro-tumorigenic effects of a high-fat diet in a diet-induced obesity model, but I always refrain from using that fat causes cancer. I think it's all context-dependent, and we need to understand the mechanisms so that we can target the mechanisms for therapy. So when you think about the diversity of potential dietary patterns that humans may have, it's impossible to study these things in humans. You can study obesity or obese patients, and even in that context, there are some variations. For that reason, we developed some systems where we study this on a dish, on a lab dish. So we grow tissue structures called organoids. And so in that context, then you can study how different ratios of nutrients such as fat and sugar can influence the mechanistic basis of the of that link that has been postulated between diet and cancer, or obesity and cancer.
So have you created a series of stomach organoids? Is that what you're saying, or what kind of organoids?
So we study intestine. So we created intestinal organoids. And so we try to understand how does those nutrients influence immune cells and epithelial cells in a way that we construct this in a three-dimensional environment on a dish using organoids and isolated cell types.
But in these organoids, though, what's the microbiome profile? Do they even have a microbiome, the organoids? Because that could be a major confounding factor.
Yeah, so right now, like when you make an organoid that does not have a microbial component, but there are some studies, and we have been trying those strategies as well. You can provide some factors that you isolate from a microbiota and add it onto the organoid to understand the interaction between the microbes and the epithelial cells.
Is that what you guys are doing? I mean, do your organoids have a microbiome associated with them? Or are you just providing some metabolites of the microbiome? What's it look like?
Yeah, we mostly try to provide the factors from the microbiota, either like metabolites or some ligands that are triggering immune system. So right now we are not recreating the actual physiological interaction that can happen, but it's close enough. It's good enough that we can study it, but it's not perfect. And our goal is in utilizing these dish-based culture systems is because this will allow us to also utilize human samples, and we can rely less on mouse or rat experiments and do more humanly relevant studies.
Do you know of anyone that has created organoids and partnered them with various bacteria that would occur in the gut to see their interaction?
Yeah. So there are studies that use a different approach called organ on a chip technology, and with those, there are some studies who have demonstrated the ability of growing, certain bacteria together with the mucosal layer of the epithelium. We haven't done it in my lab yet, but we are looking into it, and it would be great actually as the technology improves to model the interaction between different cell types in our body in vitro. That would be very useful for us to use and ask the question, how does now nutrients influence those interactions? How does fat or sugar or vitamins or other metabolites can influence those interactions?
Well, have there been studies where you have two mice, one has a normal microbiome or some microbiome, one doesn't, and then you give them the same exact food, and then you see what comes out, look at their feces and see if it's of a completely different character or not, or yes. Look at them longitudinally, because, what if the microbial fraction interacts so heavily with the intestines that it's a dramatic change from what they would do without it, and then that kind of ends up, I don't know, maybe what if it makes all the research useless?
Yeah, so the microbes are drastically respond to dietary changes. So microbes during the day, they change their community composition, but they reach a balance in a given organism. So I'll give you, the best example I can give you is you should consider microbiome as a ecosystem, such as, like an Amazon rainforest. So like in the Amazon rainforest, depending on, like the season or the climate of the region, you can have different types of animals represented, or, even plants represented. And so you can change that representation by skewing the climate or by changing the input that you give to that ecosystem. The same thing happens in the dietary challenges or other challenges that you do to the intestine. Microbes drastically change. But then the question is, how much of these changes are causal to a particular phenotype that you're studying, such as, like cancer or diabetes or depression? So the answer is some of them are causal. So people have published some studies. We have some studies that are going to publish soon. We can identify some causal links where microbes can influence, the changes in microbes can influence the phenotypes that we are observing. But most of the time, these changes are just correlations. So you can find statistically significant microbial alterations based on any input that you give, but finding a causal link is not that easy. You can even find a microbe that correlates with the type of jacket that you wear every day, or like the way that you, if you run or if you don't run to work, it can change your microbiota. But we don't know how much of these changes are going to affect causally a phenotypic outcome.
I guess, my worry would be that, you study the models without the microbiome, but again, they have such a dramatic influence that, what if you wasted years of research? What if the things you find just don't apply at all? It's like studying a mouse, and then once you get to a human, it's so different that the drug doesn't work. I guess.
Yeah.
It would just be it would be a concern.
Yeah. So that's a very valid concern. But I think it would be important to also know that in science, we do sometimes reductionist work where we take individual modules and we study the mechanism, and then we go back and confirm whether or not that mechanism that we uncovered holds true within the in vivo complexity. So in all of our studies, we try to do that. So we study the mechanism, let's say in an ex vivo system on a dish where it's devoid of many other variables. But then once we find a mechanistic node, then we confirm this mechanism in vivo. And so that is true both for in vivo, meaning that inside the body for mouse-based experiments and for human-based experiments. But of course, every approach has its own limitations, and the best scientific discoveries come from evaluating those limitations and finding what is really matter and what the truth is. And so in some of our phenotypes, they are influenced by microbes, but some of them are not. So it's a matter of finding in what sense microbes play a critical role. If they are playing a critical role, then avoiding microbes in our studies will confound those studies for sure. But we always trace back and check back and confirm that our phenotypes that we are studying, such as risk of cancer or stem cell activity or immune activity, is confirmed within the organism, within all these complexities of the organism.
So what about the cellular signaling? In the same tissue type, okay, I could see an organoid would emulate that, but it seems to me there's tremendous communication between the gut and first receiving signals from the mouth when you first start eating. The brain. I mean, that all seems to be left out of the equation again. How do you incorporate all that and see what that does?
Yeah, so answering this in one experiment is impossible. So that's why we take a modular approach. So my lab has expertise in stem cell biology and immunology. So we can do reductionist work that can focus on particular mechanisms within individual compartment of cells. So for questions that are targeting stem cells, we mostly use organoids to model it. For immune cells, we do some immune activation or migration assays. For microbiota, you need to use it from the host organism. So we try to do some sequencing experiments and try to understand how the composition or the abundance of bacteria and microbes are changing. But integration of all these modules and components is the challenging aspect. Because in science, as I told you, you need to zoom in and be reductionist to uncover one thing. You need to be very, very focused, but that super laser focus sometimes forces you to neglect other contributing factors in a tissue. So we are hoping that in the next maybe 5 to 10 years, with the advancement in profiling technologies such as single cell gene expression profiling, single cell metabolite profiling, we will be able to get a cell-based resolution of these interactions and how these interactions are changed in response to diet and nutrition and other diseases. But right now, it's very, very difficult because when you look at a tissue, this tissue has probably, multiple different cell types in different states, and they can respond to different signals. And you cannot measure all the signals at the same time right now. You can measure one by one or maybe 2 or 3 of them in one cell type. But in a tissue, let's say you have 20 cell types that can be critical for the function of that tissue. For example, in the intestine, you have goblet cells that can secrete mucus. You have absorptive cells that can absorb nutrients. You have Paneth cells that can secrete antimicrobials. And so, you cannot study all these different cell types in one experiment. You need to study them separately. And some laboratories spend, their entire career on focusing on just one particular cell type and how certain signaling molecules influence that particular cell type. So, long story short, I think in the current state of scientific knowledge, we are getting hints on how different components of cell types interact and how different signals regulate these cells, but we don't have a comprehensive understanding. So in the next 5 to 10 years, with integration methods and that involve computational biology and also profiling methods that can account for like single cells, we will be able to get that level of resolution.
Well, what have you elucidated? What major concepts or really interesting things have you discovered with your current models already so far?
Yeah. So before I came to Cold Spring Harbor Laboratory, I studied how immune system develops by the help of certain epigenetic enzymes, which can help turning on and off certain genes in a given cell. And so that was a important discovery for us to understand how certain cells can acquire functional and phenotypic states, because you share the same DNA in all your cell types, but the way that you utilize that DNA is called epigenetics, and it helps, for you to have distinct cell types in your body and distinct cell states in your body. So in immune system, that's a very commonly observed feature. You have immune cells that have lots of different functional characteristics. And that there are probably cells that can engulf some bacteria. There are cells that can react to it by secreting some cytotoxic molecules. And so the discovery that we made was focusing on a particular cell type that can recognize some lipid antigens, and we wanted to understand how specifically these cells develop by opening and closing certain parts of the DNA. So that was one discovery we made to understand how immune cell states are governed. And then the second important discovery we made was using this diet-induced obesity as a model. How does high-fat diet contribute to tumor formation by influencing stem cell biology? And we found that fatty acids can activate a specific transcription factor that senses those fatty acids in the stem cells and then make those stem cells hyperactive. And this hyperactivation, when it's coupled with an oncogenic mutation, can increase risk of cancer in the intestine. And so now in my lab, we are building upon these observations, and we are working on understanding how does this link between diet and cancer, or obesity and cancer, can be studied in an integrated way. And in that context, now we have found that when you perturb the system by the high-fat diet, the changes in the microbiota affect the way that immune cells, such as T cells, survey tumor-initiating cells in the gut. And so that's now building upon our previous observations, now connecting different modules, stem cell module and immune cell module, and then using microbes also as a separate module in understanding how cancer risk can be affected by diet. And the ongoing investigations are focusing on how immunity is changing, what are different immune cell subsets that are responding differently to different types of nutrients, and we are getting some interesting results that can hopefully be useful for understanding and treating cancer.
Well, what can you talk about? What kind of interesting results are you seeing? You said that high-fat diet, more prevalence of fatty acids, those seem to turn on certain oncogenic functions. Any other interesting insights?
Yeah, so in the context of immune cells, as I mentioned to you, we find that when you give high-fat diet, which is one type of high-fat diet, which is lard-based, you downregulate certain molecules that allow immune cells to recognize tumor-initiating cells. So that recognition is a fundamental mechanism that the immune system utilize to find and destroy a threat in our body. And we found that a high-fat diet dampens that mechanism by certain mechanisms that involve microbiota. So to go into more the specifics, we find that the diet that is high in fat depletes certain microbes, and these microbes are necessary to an extent in maintaining the immune recognition molecules on the epithelial cells which initiate the tumor. And then when immune recognition is not proper, immune cells cannot respond and clear the tumor. And we found that if you have a epithelial cell or epithelial tumor initiating cell that is devoid of this immune recognition molecule on their cell surface, they can give rise to much more tumor formation. So that's a link between basically diet and cancer that involves tumor immune recognition.
Interesting.
Yeah.
Okay. Yeah, I guess there's so much to figure out. It's kind of mind-boggling. What would be a happy result for you to have in the next couple of years with your studies? What do you think is possible to figure out?
Yeah, I mean, my biggest passion is to understand how does the nutrient as an input, since the moment that you start eating it, digesting it, releasing it to individual cells, how does this process influence the molecular and cellular mechanisms that define what we call health and contribute to disease states? Because we don't know it. So I want to create a blueprint of all the changes that can occur in response to a nutritional perturbation. And so this is a very difficult thing to do. I know all the complexities because there are so many different signaling pathways and so many different cell types that can be altered. But as I mentioned, if you do a reductionist modular approach while keeping your mind open, I believe that's going to be doable. So, dissect individual cell types and individual modules piece by piece, and then just recreate the puzzle. And so that's been, so far relatively successful, and we are hoping in the next few years we'll be able to make impactful discoveries using that strategy.
Okay, well, very good. What's the best way for people interested to dip their toe in to start finding out about organoids, about various cancer studies, etc.? What do you recommend they start with their interest?
So for nutrition related stuff, I would recommend them to follow American Cancer Society or CDC or FDA guidelines. If they can find them, I think, online. I would refrain from following any hyped up, non-scientifically generated claims online. So those are very concerning to see as a scientist. So there are lots of big claims out there. If you go research about how diet influences your health, I would suggest people to try to find the primary literature. I know it's very difficult to understand all the scientific terms and stuff, but if you read few papers here and there and then try to look at more popular science articles that are backed by scientific literature, that can be helpful for organoid-based studies. Now this is becoming more and more popular in the academia. So historically, people used cell lines, which are grown as 2D layers on dish. And now many labs are utilizing organoids to model disease and study cancer biology and other disease modeling strategies. And for that, I think American Tissue Culture Collection is gonna have some informational tools. And so individual laboratories like my lab and other labs around the world, they have some informational websites and videos. So I would recommend finding those, but making sure that these are supported by primary literature and not some like hyped up big claim that is not supported by scientific studies.
Right. Okay. And what's the best way for folks to find out more from you, read papers from your lab, get in touch, etc.?
Yeah. Well, I'm not very active on like social media, so I don't have Twitter or Instagram or whatever social media outlets out there now. I have a lab kind of profile, and then I have an email address that I use. So I'm still, learning how to utilize the technological advances in disseminating science. But yeah, my email address probably will be the easiest way to reach out. And of course, as I mentioned, so scientific publications are always deposited to a public database called PubMed for biological and medical sciences. It's www.pubmed.gov. And so like people can find articles in PubMed. Google Scholar is another database for papers. Yeah, like my email address is beyaz@cshl.edu, and if anyone has any question or would like more information about our research, I'll be happy to respond.
Okay, well, very good. Well, I appreciate you coming on the podcast.
Yeah, it's great. Thank you so much for hosting.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.